The next speaker is Samuele Marro, who has two roles within Mount Sinai. He runs a very exciting laboratory working on Fragile X syndrome and using organoids, which you heard a little bit about from Anna before this. He also runs the Mount Sinai iPSC stem cell core facility. At various times, we're proud to had been some of the major users of the facility at the Seaver Center. He brings a lot of expertise and gives access to everybody at Mount Sinai and even to a broader community, including rare disease family foundations, access to this revolutionary technology. Originally, Samuele was going to speak in the morning, and it could have been a good intro to what Anna presented, but Samuele was quite busy this morning taking his citizenship examination, which he passed. So for all of you who are naturally born or citizens by birth in the U.S. and you know nothing about, like myself, about all the things that citizens by naturalization need to know, you can ask them questions about the three parts of government and all those sorts of things that we barely can keep straight. So congratulations on that. And also much appreciate your important work at Mount Sinai. We brought him in to create this critical facility that we can all use. Samuele, looking forward to your talk. Please welcome Samuele Marro.
Okay. Thank you, Joe. As Joe said, if anyone has questions about Eisenhower, I will be happy to answer that after the talk. But now we can focus on stem cells. So it's a great pleasure to be here. I have to say that it's really refreshing for a person like me that spends most of his time in the lab to hear about all these different approaches to study neurological disorders, and really gives us the different ideas and a better perspective to what we are doing every day. So today I was asked to this quite complicated task to convince you that iPS and stem cells are important in in this kind of research. So I'll do my best taking you for a tour in our scientific lab and to tell you a little bit of what we do every day. So before I start, I would like to answer the main question: why do we need the pluripotent stem cells? And I hope the answer is partially in this slide. I always like to show the human brain and to say how complicated really of a machine is the human brain, and how difficult is to access. It's so different from any other organ, right? So if you very few people are okay in taking brain biopsies, but even if we're lucky enough to get a little bit of brain tissue, what we get out of it is cells that are not functioning and are not alive. So we cannot really study the function of neurons, human neurons. And so historically, for many years, people have turned to mouse models, which have been fantastic, as we heard in the previous talks. But as you can see, there is a huge gap between the brain of the human and the brain of a mouse. And that is simply because there are 90 million years of evolution in between the two species. So it's very important to have access to human neurons and study all the genetics that we heard about in the proper cell type. So to do that, iPS comes to help. I'm sure everybody has heard about iPS here. The Shinya Yamanaka has won a Nobel Prize recently to discover or to invent in this technology, really. But I want to remind everybody that iPS is a technology that can take any somatic cells from your body, skin cells, blood, recently even from urine we've been making iPS, and it can turn it back in time into a pluripotent stem cells. And what is pluripotent stem cells? Well, the definitions are here. Pluripotent stem cells are important because of three major features. Number one is self-renewal. They can basically make copies of themselves indefinitely. They are truly immortal cells. The second is the pluripotency, the ability to become something else, to differentiate virtually in every cells of the body. And the last feature, which I think is very important, is their ability to have their genome modified by CRISPR engineering. And so when you put this all together, you can understand how we can now start from a patient, so patient with a disease, any disease, really, go through a fibroblast or blood cells, generate IPS, and then finally differentiate and generate the different cell types that we think are important for the disease and study the mechanism in the patient-specific cells. So when you put this, organize this in a flow, you come up with what I like to call the neurological disease in a dish model, which has different steps. So the first step is, as I said, starting from the patient, and again, the power of IPS is that they come in from the patient, they recapitulate the entire genome of the patient. So you have access, you have a snapshot of the genome. Sometimes you know the genetic problem, so you can even go with CRISPR. We have this tool in the labs. We can go with CRISPR and correct the mutation, or you can insert the mutation in a normal cell line and create what we call isogenic cell lines that share the entire genome except the mutation. And then you move to the next step, which is differentiation, right? So you can take this IPS, modified, create the cell type that you think is important for the phenotype, and then move to the third step, which is probably the most exciting. Try to understand what is wrong, what this mutation is really doing in the cells, and what we call phenotypic assay. You try to find a phenotype or a difference between the mutant and the normal cells. And that's, as I said, this is really the most important step that we do in the lab, which opens up for the last step, which is the scaling up, the high throughput assay based on this phenotype that we defined, that can allow the screening of compounds, everything we heard so far, gene therapy, drugs that can revert the phenotype and can ultimately go to what we all try to find here, a cure for the disease. So to get you into this, I thought I would present two different stories. The first one is strictly about ASD, and I think it's important to highlight the value of IPS in detecting human-specific phenotypes, things that we would have never seen using animal models. And so the story is about neuroligands, in particular neuroligand 4. Neuroligand genes family are a very well-known family of protein. They are expressed in synapses, which is where the neurons make connections. And Joe can correct me if I'm wrong, but neuroligands are probably one of the first genes that were discovered in ASDs. The mutations are very rare. Fortunately, there are probably 50 or so mutations found in the whole world. But the importance, the reason why we think are interesting is because they have almost 100% penetrance, meaning that if you have a patient with a mutation in that gene, you have ASD, almost for sure, right? So we think it's a very rare but important paradigm to learn what is going off molecularly in the ASD. There is one little problem, that most of the mutations are found in the neuroligand 4, is a family of genes, right? But most of the mutations are found in the neuroligand 4, and the neuroligand 4 is essentially not found in animals, in any other organism. There is a neuroligand 4-like, but as you can see, when we look at the protein sequence, it's very different from the human. And so we thought that this would be the perfect case of see if we can learn something about the human-specific gene and compare to what we learned so far using the animal models. So what we learned so far is this, that the neuroligand 4-like in the mouse is expressed in the inhibitory synapses, is expressed in the brainstem, is actually expressed at a very low level. We don't even know where in the genome is. No one has been able to map it. But basically, we started from this. And then we turned to IPS. So we did exactly what I showed you before. We started from a normal, in this case, wild-type IPS. We used our CRISPR-Cas9 to introduce the mutation that we wanted to study. We went for the basics, right? When you want to understand a function of the gene, you just break it. So we made a knockout. We also went a little fancier. We introduced a patient-specific mutation, this R704C. That is the mutation found in the patients. And we also tagged the gene. We put a little flag so we could see where the protein would go. And then we will apply our protocols that we have established in the lab to differentiate, to go to the next step, to differentiate from IPS to different subtype of neurons. Specifically here, we made GABAergic and glutamatergic neurons. What we found, so basically for the first time, we could see where the neuroligand for human protein was. And we could confirm, this is using the HA line, we could confirm that it goes in the synapses. So this is first time anyone has seen a human neuron in real time expressing the neuroligand 4. Not only that, we also look at different expression in different types of synapses, inhibitory synapses or excitatory. And here's where the first surprise came. I told you in mouse, this protein is mainly important in inhibitory synapses, but turns out in our human neurons, it's actually mostly expressed in excitatory synapses. We also did some super resolution, and we confirmed that it's nicely overlapping with the PSD95, which is a marker of the postsynaptic side. So we could really pin the protein expression. And then we took it a step further. We wanted to ask, are these neurons with the patient mutation or with the null mutation different in any way in their activity? And so we generated what we call mixed culture of both inhibitory and excitatory neurons, and we inserted our mutant neurons in the culture. And again, another surprise came here. Well, two surprises. The first one is that knockout, taking out the gene, actually doesn't do much. The neurons are completely fine. But when you have the point mutation that you find in the patients, nothing really happened in terms of total number of inhibitory synapses, which is what you would expect based on the mouse model, but we see an increase in the number of synapses in the excitatory compartment, so that's completely new. Similarly, when we did patch clamping, which means measuring the synaptic currents between two neurons, we did not detect any difference in the inhibitory synaptic currents, but we detected a very specific phenotype in the excitatory. Again, making sense with everything we have found. And then, as usual, when you observe something in an IPS model, which is different from the mouse, you start to wonder, well, is this really reproducing the real neurons in the real brain? So we were lucky enough to find some human brain tissue post-mortem. And we probed for an antibody that only worked for Western blot. We probed for expression, and we observed that it indeed more expressed in the cortex. So again, correlating with what we found in our IPS. So I hope in this first part, okay, here's the first conclusion. We show that the human neuroligand 4 has completely distinct feature from the mouse. It has expression in the cortex, which is, again, probably a region more involved with the autism phenotype. It has, when we have the patient mutation, the R704C, it gives a change in the synaptic activity, which is exactly the opposite of what you find in the mouse. I didn't show you here, but believe me on that, when you put the same mutation in a mouse, you get exactly the opposite effect. You have a decrease in the synaptic current. So also the phenotype is very different between the two models. And then in general, I hope this convinces you that the IPS neurons are a very powerful way to detect human-specific phenotypes. So with the little time I have left, I'd like to switch gear and move to another cell population. I know normally when we think of brain, we think of neurons, which are probably the most known cells in the brain, but there are many other cells, including microglia that are recently becoming an interesting focus in my lab. So microglia is an essential cell type in the brain, is involved in many brain function. You can think of microglia as the housekeepers. They monitor the brain and they do the cleaning, removing debris. It's basically the resident macrophages of the brain. They come from a completely different embryonic origin. They come from the yolk sac. So very early in your life, you had this microglia moving from the bone marrow, getting into the brain, and then being trapped there. Once it's there, it will live the whole life in your brain. The idea is that doesn't really leave the brain. And when it's in the brain, it does a different function that include the phagocytosis, controlling inflammation, and really fine-tuning the synaptic connection between neurons. And so we got interested in this population, and our lab, together with other labs, developed a protocol to make microglia from iPS, what we call induced microglia. That is a similar protocol to what we use to make neurons that express the typical human microglia. So we can basically have, for the first time, human microglia in a dish. And we decided to study for a quite interesting project, which is involving clonal hematopoiesis. So for this, I will need to take a step back and introduce you to clonal hematopoiesis and the reason why we decided to study it. But I think if you bear with me, this would be an interesting example of a use of iPS derived cells. So clonal hematopoiesis essentially is a very common disorder. I'm sure many people in this audience have clonal hematopoiesis and they don't know, because usually doesn't give you any major phenotype. Is essentially your bone marrow starting to produce mutant cells with mutation in this DNMT3, TET2, and SX1. And these cells, little by little, they have a little advantage and they take over your blood. They become clonal. As I said, it's probably not a major disease, but the interesting thing, at least for me, is that has been associated with every single disease you can think. As you can see, it's associated with more chances of getting tumors, more cardiovascular disorders, more myocardial infarction, heart failure, basically you name it. But our attention got triggered when this paper from Stanford was published, where in the genetic studies, they observed that CAH, as I said, correlates poorly with everything, but apparently protect from Alzheimer's disease. Again, it's a little change of topic from autism, but I thought that it's another example of neurological disease that can be very interesting for this audience, and it's an example of using iPS-derived cells. And so we thought, well, we have our iPS-derived microglia. Maybe we can try to understand why the carriers of CAH mutation are somehow protected from Alzheimer's. And so we went back to our model, again, the same strategy using CRISPR from a normal iPS. We generated this very specific mutation that you find in most of the people in the three key genes, SX1, TET2, and DMT3, in heterozygosity. And then we applied our protocol to generate human microglia with the CAH mutation. And this is all preliminary, it's unpublished data, but so far we have observed some difference between the mutant and the control cells. And interestingly enough, we observed that all the mutants have more propensity in cleaning up debris. Specifically, one debris, which is called amyloid beta, I'm sure you have all heard, it's very important for Alzheimer's disease. So it seems like the mutants are better in cleaning the A beta. Not only that, we also do single-cell RNA-seq, and we found that the mutants have a gene expression profile that correlates more with what is called DAM, which is a disease-associated microglia state. I don't want to go into much into the details. I just want to leave you with the message that microglia is, as I said, does this very important function of cleaning, removing synapses, and removing dead neurons. But sometimes when it's overactivated, it just does it too much, it can become a problem, okay? And so we think what we observe could be an explanation of having an hyperactive microglia that is able to clean the debris better, maybe in a part of the disease, and therefore correlating with the Alzheimer's protection. The one last thing that we are also doing that I thought would be interesting for this audience, we are taking this microglia, which is not really something you can do easily with iPS neurons, but with iPS microglia, you can put them back in a brain, in a mouse brain. So again, all these IPS models are fantastic, but they are isolated in a dish. In this case, we are putting them back in their environment, in a brain environment. And so what you can really have, you can observe human microglia acting in their normal environment in a brain, and you can ask whether the mutations are doing different things. So we also think this is a very powerful application of the IPS cells. So these are the conclusions for the second part. I showed you that the mutant CH microglia has enhanced phagocytosis. It's just better in cleaning amyloid beta. We observe a different activation state in the mutant microglia that can be correlated with the disease. And again, in general, the message is similar to the first story, that IPS cells are probably the best tool we have to study human mutation in the human genome in human cells in the context of active human cells. So with that, I'll leave you with work in progress, which is not only from my lab, from many other labs at Mount Sinai and outside. These all IPS models are really moving to be used in studying neuronal properties, the synaptic activity, and study the network balance between different activities. The other major focus is the gene expression and understanding the chromatin profile that is affected by patient mutations. And finally, as you heard already from Anna, there is a lot of work in developing 3D system that can recapitulate what some people call mini brain, recapitulate the early brain developmental stage that are again very different from what you find in a mouse. And really, all of this, again, I want to remind you that the final goal is to close the circle and go from patient to a phenotype and ultimately develop a drug or a cure for any kind of disease. So with that, I'll stop. I'll just say a big thank you to everybody in the lab, specifically Madel, Ruichi, Erdin, and Laura. The Papa Petru lab, that is a collaborator with the Alzheimer study, and of course the Seaver Center, Joe and Anna, for the great discussion and collaboration all the time. And I'm not sure if we have time, but I'm happy to take questions on IPS or anything else.